What? Madison, did I lose you? No? Okay. I got a message that said I might lose some people and your name is on the list. I don't know why. All right. So anyway. Okay. No, that's fine. I just didn't want you to get kicked out of the meet because I was trying to. All right, excitation, contraction, coupling. Again, that is this idea that the nervous. Okay, the nervous side versus the nervous side until the idea of this sarcomere contraction. All right, so it is a series of 6 steps. They are nice and pretty on page 329. All right, we're going to start with the top. All right, the top is neural control. So this is everything we learned about the neuromuscular junction and action potentials and membrane potentials. So the neural control is the start. So this is where the neuron is going to come up close to the sarcolemma, which is the cell membrane of the muscle fiber. This is going to start the process of, yeah, I think one of us does because just Julia, can you hear me okay?
Yeah, I can hear you fine.
Okay. Okay, so the neural control is again action potential coming down to the axon terminal. It starts everything that happens at the neuromuscular junction. All right, the next step is excitation. Okay, this means that an action potential is causing the acetylcholine to release, and it leads to the action potential spreading to the sarcolemma. Then we have the calcium ion release stage. All right, so remember that excitation, not only does it go through the sarcolemma, but it also travels through the T tubules, which are continuous with the sarcolemma, and then to the sarcoplasmic reticulum. Okay, and it's in these wide areas of the sarcoplasmic reticulum called the terminal cisternae, where the most of the calcium is stored, and that is what gets released. And we know that calcium plays a huge role in the sliding filaments. The next stage is the contraction cycle begins. So the calcium, again, plays this role. It binds to the troponin, and then that tropomyosin cord is slipped out of the way of the active sites on the actin. That allows the myosin heads to actually form a cross bridge as long as ATP and calcium is available.
Miss Boutreau.
So this contraction cycle continues. Yes.
I can't I don't know, it sounds weird. Like I can't really hear you. Like it's like keeps making a funny noise every time you talk.
Did you? Okay. Did you try and restart?
How do I do that? Do I just click out and click back in?
Yeah. Try and restart it.
Okay.
Let me just wait a minute here. Technical difficulties. I wonder if this had something to do with that recording being so slow. Are you back? Can you hear me okay?
Yeah, I can hear you.
Oh, perfect. Okay, you sound better too. All right. So the contraction cycle is this idea of the cross bridges forming and the actual shortening of the sarcomeres. So they will continually shorten until that is used up. The ATP and calcium is used up. I think of step 4 and 5 to kind of together, because it's just going to happen continuously. Stage 6 is tension. Okay, so tension is this idea. So tension is a big idea. Okay, this is not happening at the sarcomere or at the muscle fiber. Tension is happening at the organ level. So if you think of your biceps has this big belly part. Actually, there's two. There's this two big belly, which is kind of like the big meaty part of your biceps. But then they have tendons that are pulling on the lower part of your arm and your shoulder, okay, your humerus. Where the tendons are being pulled, or when the tendons are being pulled by the biceps because you're contracting it, that's the idea of tension. So tension involves the muscle organ and the tendons and the bones. Okay, so the technical definition in the book, a pull or tension on the tendons at either end. Okay, so muscle tension, again, is this big idea in the muscle. All right, I am going to refer you to Post-it note, page 329. Because page 329 is like the big picture. You know, we've been talking big pictures yesterday and today of the entire process. And page 329, if you see a piece of that whole picture that doesn't make sense, that's when you flip back to section 1 and you study the structure. Maybe you don't know about the neuromuscular junction. So maybe you go back and study the structure of the neuromuscular junction, and then what happens, the physiology of the neuromuscular junction. Does that make sense? All right, so take a minute on 329 and just look through the picture. Would you like me to go over any part of that in detail again, or maybe briefly, whatever? Is there anything that doesn't make sense that maybe you wanna focus on again?
I missed excitation because I couldn't hear you.
Okay, excitation is the idea that the action potential that has been, the action potential that it was in the neuron, it released the acetylcholine, and the action potential then spreads to the sarcolemma of the muscle fiber. So it's that acetylcholine that's kind of connecting the neural side to the muscular side. All right, anything else from this picture as a whole that we should look at again?
The calcium ion release.
Right. So the action potential travels to the sarcolemma of the muscle fiber, which is the muscle cell. Okay, and the so since the T tubules are continuous with the sarcolemma, the action potential is actually going to travel down the T tubules till it hits the sarcoplasmic reticulum, and that's where calcium ion is stored. So calcium pumps are going to start releasing calcium. Okay, calcium is then going to play a role in the sarcomere contraction, and that's the next step. So another way we could, like the title of this page is excitation contraction cycle. Excitation is the neural part. Okay, that's happening. The action potentials traveling, propagating. The contraction is the muscular part. So this cycle is what describes what's happening from the time there is a message sent to contract the muscle to the time that the muscle is actually contracted. All right, we're going to move on to tension and twitches. The rest of this chapter really talks about sort of how things work together, how muscles work together, and how tension is produced, and how a muscle might twitch, and you know the different levels of contraction. So if you're having a tough time kind of visualizing it, you know, let's stop and like rethink where we're at. So stop me if something doesn't make sense, and we'll kind of put it back into perspective of the muscle fiber or the muscle organ or what, you know. All right, let's start with, if you would flip the page to 330, that, if I haven't said this already, this book has really, like, the first version of this Martini book that I used was horrible. It did not have all these nice, pretty pictures with the boxes of information that kind of go with it. It was just text and a few pictures. And so this has really evolved into something that's very student friendly. So having said that, the picture on 330 does go through some of this tension ideas and twitch. All right, so tension is greatest when muscle fibers are stimulated at optimal lengths. Well, what does that mean? That means that muscle fibers are either on or off. You're either producing tension, which is what we think of as a contracted muscle, or they're not. Okay, they're off. So the optimal resting length of a sarcomere is when you have the optimal number of cross bridges that are potentially able to form. That means the potential to produce the most tension, right? So if you think about that for a second, yeah, the more cross bridges that I can form in a single sarcomere, because there's hundreds of myosin heads, the more efficient that contraction is going to be at the sarcomere level, at the muscle fiber level, at the organ level, all right? So the maximum cross bridge formations. However, we do have things that limit this. So just like we have something that limits, for example, the size of the cell, we have things that limit the maximum tension. And that is muscle arrangement, connective tissues, and bones, so that we don't want to overstretch or overcompress or overcontract any of these muscles. So the limits are placed on there by other muscle or other organ systems. All right, so we're going to go through this. We're going to go through this picture before we start talking about twitch. So if you look, there's a red start button. Wow. Start arrow. Okay. So this is demonstrating optimal lengths. Okay. And if you look at the graph, remember we talked about, prior to this, we talked about like repolarization, depolarization. This is different. I mean, the graph looks similar because, you know, you see a peak, but this is different because this has to do with the length of the sarcomere versus the tension on it. Okay. All right, so the first one is the optimal range, okay, the optimal range, and that's the peak. And if you look at the bottom here, it says optimal resting length. The normal range of sarcomere is about 75 to 130% of the optimal length. So that's kind of your peak. And then as you go down either side, it's kind of talking about the conditions that you would see the sarcomere at. So let's go down the left side first. A decrease in the resting sarcomere reduces tension because the stimulated sarcomeres cannot shorten very much. And so what you see there is not a whole, or I'm sorry, it's actually looking shorter, correct? You see that it's kind of looking shorter on this side. Then if you continue down the left side, you'll see no tension, and these things cannot shorten any longer because of the Z lines. So your tension is 0. So that's when you kind of overshoot the shortening. If you look on the other side, okay, you can see if you overshoot the lengthening. Okay, so you see these lengthening. So here as we go down, reduce the zone of overlap. See, this is the zone of overlap for the actin and myosin. So there's fewer cross bridges that can form. So then we come back down to the 0 zone of overlap, so there's no tension at all. So on both sides of this, if you overshorten, you can see there's no tension because the Z lines are so close and there's like an overlap of the overlap zone at the M line here. So those cross bridges won't form, and so you have no tension. On the opposite side, if the actin and myosin aren't touching, this is over lengthening, then you can't produce tension on that side either. Okay, so optimally, you have the myosin heads and actin active sites at maximum, right here. Does that make sense? All right. So if you think about a muscle contracting for a second before we even look at this set of graphs, you have certain phases. Hopefully I have this phase. Here we go. You have a stimulus, which comes from the nervous side. Then you have the phase where it's contracting, and then you have the phase where it's relaxing. Okay? A single stimulus, contraction, and relaxation sequence is called a twitch. All right? You can see that twitches vary in time. Something like the eye muscle, which if you think about your eyes and how quickly they react, it's a very, very short twitch. Versus your soleus, which is behind your calf muscle. Your gastrocnemius is actually the calf muscle. That's a little wider, but the soleus is a deep muscle, and so that's got a very, very wide, long twitch. Right? So the duration of that twitch varies by the muscle type, location, environmental factors. A fasciculation is an involuntary muscle twitch. I don't know if you guys get this, but my eye muscles do that all the time. And I've read that it has something to do with the amount of caffeine that you intake. Well, that's probably true for me. Okay. So anyway, if you ever get that kind of twitch under your skin, that's called a fasciculation. A motor unit is, so now we just have some terms as we move forward in this section because they're going to use it. A motor unit is a group of muscle fibers that are controlled by a single motor neuron. And we're going to talk about motor units again tomorrow. And then a myogram is actually showing the development. So this is a myogram. So showing the development of the contraction. So this is just kind of showing you some overlap with some of the physiology of what's happening with each of these. So the latent period, you have an action potential stimulating. no tension, but calcium is starting to be released. The contraction is when you actually start the cross bridge cycling. And relaxation is when your tension returns down to resting levels. So that kind of coincides with this, that latent contraction and relaxation. And that picture is at the bottom of 331. So these two charts are just sort of general information about muscle twitches so that we can talk about specific levels. Are there any questions about these couple charts or couple slides from 331? All right. If we flip to 332, this is where we're going to end today. And this is where I'd like you to please make sure you have that levels of peak tension pulls up. Okay. You're going to actually, and if you have the, it might be helpful if you have the chapter 9, what am I trying to say? Slides up too, because you can copy these pictures that are on each of these slides. So I'll give everybody a second to pull both of those up. But what we're going to do is we're going to talk about the level. We're going to give some descriptive items, and then you're going to copy the picture. It'll be much smaller. You'll have to shrink it for each of these. And if you aren't able to do that right now, then you can just add the pictures later. Fine. All right, peak tension. Peak tension in a skeletal muscle is determined by two things: the frequency of the stimulation. So how frequent do you see a stimulus? And on this page, you will note a stimulus is a green arrow. And the number of muscle fibers stimulated. So you get peak tension when you have more stimuli and you have more muscle fibers being stimulated. Okay? And so there are several types of this peak tension. And they are on 332. We're going to start with treppe. Okay, treppe is when you have relaxation. So imagine, before we even talk about this, imagine like one of these being that latent period contraction and relaxation from the last page. Okay, so now we're talking about, let's put a bunch of those together to demonstrate tension. Okay, so the green again is the stimulus. So you have a stimulus right after each relaxation phase. So we have the latent period, we have contraction, relaxation, another stimulus, contraction, relaxation, another stimulus. So it looks just like this slightly growing set of waves, like a staircase, but it's very gradual. Now, treppe is found mostly in cardiac muscle. And not skeletal muscle. If you think about skeletal muscles, we don't need this type of continuation with most of our skeletal muscles. We need more certain times. You know, when you're standing up, you're only using a certain amount of your muscles. So that tension needs to build immediately and as we need it, not continuously like cardiac muscle. Okay, we're going to move on to the next type, which is wave summation. Okay, and this is kind of the continuous addition of twitches. So your stimulation before you do, you want to write it
One
down?
mouse keeps like going away.
Oh, is that happens to my Mac a lot lately.
So
So if you look at the difference between wave summation and trepe, you can see a full like relaxation contraction and up and down to the waves. But with wave summation, you don't see that full relaxation. Before it hits the bottom of the relaxation phase, another stimulus hits, and so then you have contraction again. So the stimulus is before relaxation ends. It's a powerful contraction. But you can see where the summation comes from. It's slowly increasing until the stimulus stops, and then you get your full relaxation. All right, next one is incomplete tetanus. I don't know if you've ever heard of a tetanus shot, but the disease tetanus causes convulsions. And so tetanus is used here because you get this convulsive tension. So this is rapid cycling of the contraction and relaxation. And you almost hit maximum tension. But because you hit some form of relaxation, it's called incomplete. And if you look at the difference here between this one and the last one, you can see that the peaks increase and then you kind of plateau at a point. And that's a big difference between wave summation and incomplete tetanus. If we just sort of stopped the graph right here in the middle, it might look like wave summation. But then you get to this point where you're almost at maximum tension and you still allow the fiber to relax a bit before the next stimulus. And then we have the last one is complete tetanus. And you can see this one looks very different. This is a high stimulation frequency. So you can see way more green arrows here. So that means that there is never a chance for the calcium ions to be pumped back into the sarcoplasmic reticulum. There is a continuous supply of calcium ions. And so you get this very smooth looking graph, okay? The stimuli are so frequent that there's no relaxation. And really what's driving this is the action potential from the nervous side. So as soon as the action potential ends, that's when this relaxation can occur again. This is not going to be found normally. Complete tetanus is not a normal occurrence. You never want to reach the maximum tension.